pleasure to introduce Christina Kudanglu, who is a research and teaching associate at the School of Basic Sciences at EPFL, and of course the CIBM Research Staff Scientist and 9.40 MRI Operational Manager and a leading member of the CIBM. So it's my great pleasure to introduce you and to introduce Dunja.
Yeah, the speaker.
Yeah. Nice to see you all, at least here in the room, and nice to see part of the people online. So as you heard, I'm Christina, and it's really a huge pleasure to introduce today's speaker, who's Dunja. Why? Because I worked with her in the past 4 or 5 years, I think, and because she's amazing, of course. And so Dunja did her studies at the University of Split, where she finished her master, but she also had her master here at EPFL, where I've met her. And afterwards, she started her PhD with us, with me. I was her supervisor for her PhD. So the title of her PhD was Advanced Metabolite Mapping at ultra-high field using proton MRS, proton MRSI, and macromolecules applications in the rat model of type CHE. So as you've heard a little bit, so she was working on type C hepatic encephalopathy. So today, for today's talk, Dunja will be just presenting a part of her work during her PhD, and this is a brain regional vulnerability in the rat brain during type C hepatic encephalopathy from metabolic to cellular scale. So Dunja, the floor is yours, and hope that you enjoy. I will enjoy.
Thank you very much, Christina, for the nice introduction and for saying my title. So I have plenty up to do now. So yes, just to go quickly through the outline of today's presentation, I will first give you a brief introduction to type C hepatic encephalopathy, and then I will go through 2 parts of my research in brain regional vulnerability in this disease in both adult and developing brain, and finish with some general conclusion. So what is type C hepatic encephalopathy? Well, it all starts with chronic liver disease. And what does this mean? Well, when a person has a chronic liver disease, he has a cirrhotic liver. And to put the disease a little bit in context, so the chronic liver disease causes 2 million deaths worldwide every year, and the incidence in 2015 was 20.7 per 100,000 adults. But the disease doesn't only occur in other adults, it occurs in children as well. So in adults, it can be caused by hepatitis and other viruses. It can be a non-alcoholic fatty liver disease, or it can be caused by alcohol abuse. But in children, it's mostly caused by biliary atresia. 40% of patients develop a condition called hepatic encephalopathy between 5 years. And when it's a consequence of chronic liver disease, we call it type C hepatic encephalopathy. So basically, what is type C AG? I will call it AG in brief. So it's a neuropsychiatric disorder, which is seen in changes in personality, sleep alterations, deficits involving both cognitive and motor performances. It is important to know that the only curative treatment for this disease is liver transplantation. And after liver transplantation, adult patients usually partially or fully recover their neurocognitive abilities, but children have permanent neurological problems. So as I explained now, all the neurological symptoms of this disease are very well described. However, the underlying mechanisms are not fully understood. And to go to one of these mechanisms, we have a key player in this disease, which is a small molecule called ammonia. We produce ammonia in our intestines as a product of our digestion. And when ammonia is produced, it goes to the liver to be metabolized in the urea cycle. So urea is produced and goes to the kidneys to be eliminated. However, if the liver is cirrhotic, we cannot produce urea, and therefore ammonia stays in the blood and eventually reaches the brain. So we have an impaired detoxification of ammonia, but also other toxic substances such as bilirubin, bile acids, and etc. Therefore, we have a toxin accumulation in the blood. And in addition, as a consequence, we have a liver inflammation as a consequence of the disease, which leads to a systemic inflammatory response. So what happens when ammonia reaches the brain? So in the brain, we don't have any urea cycle. So the ammonia is eliminated in the cells called astrocytes with a reaction with glutamate, and with the help of an enzyme called glutamate synthetase, it's eliminated into glutamine. And therefore, because of a heavy load of ammonia in the brain, we have a glutamine increase. But not only this, we know that in addition to the glutamine increase, there is a secondary osmotic answer. There is also a possibility of, there have been reported the possibility of brain edema, neuroinflammation, and formation of reactive oxygen species. Therefore, to go to the motivation of our work, as I said, these neuropsychological deficits in type CAG are very well described, but the underlying mechanisms are still not well understood, their chronology and involvement of different brain regions. Therefore, in this work, we chose 3 brain regions: hippocampus, cerebellum, and striatum, which are the main brain regions implicated in pathogenesis of AG, and we decided to study these 3 brain regions. So the aims and novelty of the work were to describe the evolution of metabolic profiles throughout the disease duration in the 3 stated brain regions, to identify the potential metabolic differences between the regions, and to use a multimodal approach, combining histology with blood biochemistry and electron paramagnetic resonance to compare this brain regional, to complete this brain regional investigation of this disease. And for this, we used an animal model of the type C hepatic encephalopathy, which is called the bile duct ligated rat. So the rats are their bile duct is surgically ligated, and this leads to development of chronic liver disease in the animals, and as a consequence, they develop a neurological disease, type C hepatic encephalopathy. So this project was divided in 3 studies. So at least I divided it here for a better understanding. Study 1 was proton MRS. So we do proton spectroscopy in these 3 brain regions. It's a single voxel spectroscopy longitudinally throughout the disease evolution every 2 weeks. So we start at week 0 when we perform before video surgery, and our week 0 is our control because the rats are adults, and then we compare always to week 0, and we follow the disease through 8 weeks, performing every 2 weeks the MRS scan. So I'll just mention a little bit about the brain regions, why we chose them. I already explained, but to be in more detail, hippocampus is responsible for spatial memory and spatial information encoding. And then if you go to the striatum, it's responsible for control of motor movement, but also decision making. And the cerebellum is involved in spatial navigation, learning, and working memory. So all the regions are have these roles which are affected by the disease. And to say a little bit more about the proton MRS, so we acquired our proton spectra on the 9.4 Tesla variant magnet using a special sequence, and we fit our spectrum with LC model, allowing us to quantify up to 17 metabolites. And these metabolites, in addition to glutamine, which I already said it's very important, are also neurotransmitters, energy metabolites. They participate in osmoregulations, they're antioxidants, and participate in myelination and cell proliferation. So it's a very good technique that allows us to follow metabolism longitudinally. The part 2 of this study were histology measurements, and this is the work of my colleague Kasia, and she at week 4 and week 8, we used different types of staining to stain astrocytes and neurons and track their morphological changes during the disease. And also in collaboration with Kasia, we did a study of systemic and central nervous system oxidative stress using ex vivo EPR measurements, also in hippocampus and cerebellum, and I will also present these results. We always compare these rats to sham-operated animals. Sham-operated animals are just animals which are controls. They are operated in the same way as bile duct ligated rats, but without the ligation. So they're just opened and identify the bile ducts, so they go through the same stress as our sick animals, but they are healthy. So we are going to start with the response to ammonia increase in endosmotic stress. So we can see here the blood parameters we measure every 2 weeks. We have an increase in bilirubin throughout the disease evolution, and the bilirubin is a first marker of the disease. Basically, it's an elimination marker. So if the animal doesn't have an increased bilirubin, it's not sick. And we see here that it's very increased until week 8 during the disease. And as I said, ammonia, we measure ammonia in the blood, and ammonia is increased throughout the disease evolution. And here I will present the metabolites we measure, and I start with glutamine. The metabolites are always presented in 2 ways. Here, I don't see my, so on the right, you have a relative increase of glutamine to better see how much it increased. And on the left, here you have a concentration of glutamine with a percentage change every week. So it's a 2-way comparison. So you see, on the right, you see all the 3 brain regions together, and you can see the relative increase comparison. And here you can see every region individually and see the increase in every region. So you see here that our glutamine is increased in all the 3 studied brain regions, and the strongest increase is in cerebellum, where it reaches at week 8 plus 134% of increase. And as a response to glutamine, and this glutamine increase, of course, is highly correlated with ammonia in the blood. And as a response to this glutamine increase, we have a decrease in other osmolytes as a response to the osmotic stress. So here, for example, we have a decrease in inositol, which is highly decreased until week 8. And inositol is located in the glial cells, so it's a very direct response to the glutamine increase because the glutamine is forming in the astrocytes. We also have a decrease in other osmolytes like total choline, which is interestingly the most decrease in striatum, but also in taurine and creatine. And you can see here total creatine, which we assume that it assumes a role in as an osmotic regulator in this disease. And if you look at the sum of all the osmolytes, we can see nicely how, for example, in the cerebellum, which has the highest burden of glutamine, the osmolytes are trying to compensate by their decreasing, but interestingly in striatum, glutamine reaches a plateau at week 6 and doesn't increase further, but the osmolytes are strongly decreasing, and you can see that on the far downside of the slide. And that's very interesting because it tells us that there is a unique response in striatum, that there is something different going on in this brain region. And I wanted to just show in the spectrum that we can also see this changing directly in the spectrum. If we highlight, you can see nicely how the glutamine is in the sick animal at week 8, how the peak is strongly, very strong. So it's highly increased, and we can see a decrease directly from the spectrum before even quantifying decrease in the inositol and taurine. So then, with the help of Kasia, again, thank you, Kasia, for this work. We were following also with the GFAP staining, the astrocytes morphology, in the changes in astrocytes morphology, in the disease evolution, and why we chose to look at astrocytes, because the glutamine, as I said, is forming astrocytes, and it goes through this osmotic stress. So we wanted to see if there is something changing in the astrocytes. And if you look at the image on the right, we can see that we show that there is a decrease in the ramification of astrocyte processes. So, and there is a retraction of astrocyte processes. Basically, so there is a morphological change which occurs in astrocytes. And she also looked at neurons, and we show that there is a different response between hippocampus and cerebellum. We show that there is an increase in neuronal soma in the hippocampus in the BDL rats, but there is a decrease in neuronal soma in the cerebellum. She also looked at different things, which I'm not presenting today, but it's a very nice addition to our study. So to continue with the metabolites, now we go to energy metabolism and the neurotransmitters. We can see that as a part of energy metabolism, we see a strong peak of lactate at week 8 only in the cerebellum, which is an indicator of an energy metabolism perturbation. But also it can be that because of high neurometabolic stress in this brain region, because it's the region with the strongest changes, we have this lift in lactate at week 8, and in neurotransmitters we show the decrease in non-significant, but there's still a decrease in GABA and glutamate. And we have a unique opportunity with proton MRS to measure the small molecular weight antioxidants like ascorbate and GSH. These molecules are the molecules which react with a very fast reactive oxygen species, but still this is an indirect measurement of oxidative stress. So therefore, to complement these results, we did electron paramagnetic resonance ex vivo study. So how does it work? Well, we isolate tissue of the brain region and mix it with the EPR probe. And the EPR probe is not ESR active, but when it reacts with the reactive oxygen species, like in this case, our EPR probe reacts with the superoxide radical, it becomes EPR active, and then you can see a signal. And basically, you can follow the time evolution of this signal, and you can calculate the ROS formation rate. And basically, we do this by integrating this signal, and we can compare this ROS formation rate between the BDL rats, so sick animals, and healthy animals. And if we look at our results in hippocampus, we did this at week 6, but also longitudinally, and we can see that there is a significant increase in this ROS formation rate throughout the disease evolution in this brain region, and that it correlates with brain glutamine, but also with an antioxidant ascorbate. We did this also in cerebellum, but only at week 6, and we show a similar increase in ROS formation rate. And we also did a systemic oxidative stress study. This means that we did the same thing in the blood. So basically, we take the blood for the animal and repeat the procedure. And we showed that at week 6, we have a significant increase in the blood systemic oxidative stress. And this increase was correlated with blood ammonia. So this is our oxidative stress study. And then to sum up, I did, because a lot of parameters were measured, I implemented this status analysis, which is like a PCA type of analysis, which to provide us a general overview of the metabolic changes. And how does this work? I present here the first axis of this PCA analysis, and this first axis explains the highest variance in the data. So basically, for us, because of the way I normalize the data, it explains the longitudinal evolution of metabolites. And the metabolites which are highly contributing to this first axis are the ones which are changing the most, and which are low contributing to the axis are not changing in this disease. And if you look at the highest contribution in all 3 brain regions, up on there in striatum and hippocampus, and on the bottom is bilirubin, because we said in the beginning that bilirubin is the starting marker of the disease. But very close to bilirubin, you have ammonia and glutamine. So basically, we can say that the glutamine is driving the variance in the data set very similar to ammonia and bilirubin, and we can say that glutamine is one of the main driving forces of the disease. And this also overview was very interesting, because we can also see all the patterns that I explained in the different brain regions on this axis. So it's a nice overview of our results. So to conclude on the adult brain study, we proved that there is a differential brain regional vulnerability to the disease in the adult brain, and we isolated the cerebellum as the region with the highest burden of glutamine and more pronounced changes in the other metabolites. So we highlight its increased vulnerability. We can say we isolated glutamine, and we said that it can be considered as a first brain metabolic marker of type CAG, and this metabolic metabolite could potentially serve to assess the hepatic encephalopathy independently of the results of the neuropsychological tests. We follow the astrocytes and neuronal morphological changes, and these changes can, and we report them, and they can influence the overall activity of the neuronal network during the disease progression. And in the end, we show the importance of reactive oxygen species and their role in the evolution of the type C hepatic encephalopathy. So this was our study in the adult brain, and then now we will go to the developing brain. But just as a reminder, I'll go back to comparing what happens in the adults and in the children. So as I said, in adults, the disease is caused by different factors, but in children, it's mostly a consequence of a biliary atresia. But the only curative treatment in both cases is liver transplantation. What's the difference is that in adults, mostly the neurological problems after liver transplantation are reversible, but in children there is these are only partially reversible or irreversible, and they lead to learning disabilities in children, cognitive deficit, and mental retardation. But so basically the conclusion is that the brain in the developing brain, certain central nervous system is uniquely vulnerable to this disease. But the metabolic changes in this developing brain are not very well described, and how the different regions respond is how is it the metabolic changes are different between the adult and developing brain. It's not well described, and this was the aim of the study, to describe in the same brain regions that we did for the adult brain, to study them in the developing brain. And this we did in the same animal model, but this time the rats were operated at P21 days after birth, and this corresponds to a 9-month-old human baby. So the disease was induced early in these rats. We used the same rat model, but now we compared the metabolic profile differently. We always compare the BDL group to the corresponding age sham group to take into account the metabolic changes which are due to brain development. So there is always in parallel a BDL and a sham group, and the sham is always the control for the BDL. We use the same method and the 9.4 Tesla variant scanner in the 3 brain regions, the same brain regions, and these are the results. So we can see here that we can observe the same pattern of the disease in terms of glutamine and the glutamine increase in all the 3 brain regions. And we can see at week 6, this glutamine increase is very significant. Now, if we take this is the percentage change of glutamine at week 6. If we compare this to the adult brain, we can see that the glutamine change at week 6 is much stronger in the developing brain than in the adult brain. So basically, we prove that there is a stronger and a faster glutamine increase in the developing brain, and this change is more pronounced. So the glutamine change also correlated significantly with blood ammonia, and we can see that the pattern of the change is similar because we have the highest increase in the cerebellum and then the lowest in the striatum, as we observed in the adult brain. We have the osmotic response to glutamine increase, which is very similar to what we observed in the adult brain. But now if we look at lactate, the lactate shows significant increase at week 6 in all the 3 brain regions, which is very different than what we see in the adult brain, where it peaked only in the cerebellum at week 8. And we said that probably this peak of lactate is due to high neurometabolic stress in this brain region. So we can conclude that there is a higher metabolic stress in the developing brain earlier in the disease, which we can observe this with this lactate peak. So to conclude, we again show that there is a differential brain regional vulnerability to the disease also in the developing brain, but we also identify differences in this metabolic response between the adult and the developing brain, like a stronger increase in glutamine and significant decrease in lactate. And we prove that there is an increased susceptibility of this developing central nervous system to the insults of the disease. So to sum up, we provide detailed descriptions of the metabolic alterations during type C hepatic encephalopathy in 3 brain regions, and we identify differences between adult and developing brain. And why is this important? Because we can, using these detailed descriptions, we can identify different targets for potential treatment strategies. And some of the treatments we already tested, like started to test, like fatty treatments, or my colleagues, probiotics and vaccine. So with that, I would like to acknowledge my group, my supervisor, Christina, and collaborators, Kasia and Bernard, for all the help during my research, and also the vet team, who had a very big role in this, because of to follow all the animals and to help me during my experiments, Dario, Stefano, Alina, and Mario, and my colleagues, who are always a very good support. And with this, I would like to thank you for your attention.
I'd like to give Dunja
Thank
appreciation.
you.
Thank you
Thank you very much.
And thank you for all your work. So
thank you.